Welcome everyone to the fifth webinar of Obesity 2020, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. This webinar is titled Cravings and Weightlifting Squats, Technologies that Explore New Metabolic and Behavioral Research, and will feature Dr. Zhen Yan, Professor of Medicine at the University of Virginia, and Dr. Diego Bohorquez, Assistant Professor of Medicine at Duke University School of Medicine. Today, Dr. Yan and Dr. Bohorquez will present case studies demonstrating the use of home cage phenotyping for preclinical obesity research. And with that, I'm pleased to welcome our first presenter, Professor Diego Bohorquez. Diego, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Thank you very much, everyone, for coming today. Thank you for the invitation, and thank you TSC for sponsoring this event. So today I want to share a little bit of what has been a long-standing question in the field that has actually become not only our mission in the laboratory, but also our passion on how we study the gut-brain connection. I will go ahead and run my disclosures. And I became interested in this question of how it is that food in the gut, and particularly the gut, affects appetitive behaviors. When I was a graduate student in nutrition, I remember being very surprised that gastric bypass surgery will completely alter the perception of food in patients. And then reading the literature, I found that as early as 1952, it was known that if nutrients are delivered directly into the stomach, they actually have this reinforcing effect on decisions. So for instance, if a rat is given the choice to go left or right, and every time that it goes right, it receives an injection of milk inside of the intestine, then eventually the rat will learn to go right just based on the rewarding stimulus of the milk. But for long, it was really unknown how it is that this system works. In fact, it was unknown that the gut had its own dedicated sensory system. But in 2008, and I will say that at the beginning of the 2000s, things started to turn around for the study of neural circuits, particularly in the brain, and certainly for the gut, because some of the tools were becoming available to study the cells, the molecules, and the circuitry that were driving behaviors. In 2008, Professor Iván de Araujo, now at Mount Sinai, he found that even in the absence of taste receptor signaling, animals were able to find a solution with sugar. So when you knock out sweet taste receptors, the animal is incapable of finding a sweetener, but it still is able to find sugar, suggesting that there was another mechanism that allow the animal to detect sugar and discern sugars from sweeteners. And this happens to be within the gastrointestinal tract. And just to position ourselves, I'm going to show you here a video that was produced in my laboratory.
How the gut and the brain communicate has fascinated us for centuries. The gut-brain axis transforms information via the vagus nerve from food to feelings. Once eaten, digested food particles enter the small intestine, which is covered with a velvety layer of villi. Each villus is lined with a single layer of epithelium. This layer is made up of different cell types. One of them, the enteroendocrine cell, is unlike the others. It is our gut sensor. In addition to communicating through hormones, we discovered that enteroendocrine cells also synapse with nerves, including the vagus nerve. We call those enteroendocrine cells synapsing with nerves neuropod cells. They sense and react to their environment. They sense mechanical, thermal, and chemical stimuli, such as nutrients or bacterial byproducts in the gut lumen. Inside neuropod cells, signals from stimuli are converted into tiny electrical pulses. These pulses propagate via synapses onto the afferent neuron of the vagus nerve. Vagal neurons carry the sensory information to the brainstem, linking the signals generated inside the small intestine to the brain. The neuropod cell connection with the vagus nerve serves as a conduit for food in the gut to influence brain function within seconds. This connection is also a potential portal for gut pathogens to access the brain. This new knowledge is a foundation for designing therapies to treat disorders related to altered gut-brain signaling.
And just to clarify a little bit of the concept, these gut endocrine cells or enteroendocrine cells are specialized cells of the gastrointestinal tract and pancreas with endocrine function. They produce hormones or peptides in response to various stimuli and release them into the bloodstream for systemic effect. These signals diffuse as local messengers or transmit to the enteric nervous system to activate nervous responses. So when we found a direct synaptic connection of these enteroendocrine cells with nerves, we defined and we extended the concept of how it is that the cells communicate, and as such we gave it a new name for those cells that are forming synaptic connections. And these are the neuropod cells, a gut sensory epithelial cell that synapses with nerves to transduce stimuli in milliseconds. And this concept was built over the years based on several studies over the last 10 years, and I will just like to highlight 3 of them. One is that these enteroendocrine cells, they have these protrusions that directly contact nerves within the intestine or the colon. And these became the foundation of, and also when they are put in culture with a sensory neuron, they can recapitulate that synaptic connectivity, as you can see here in this video. So this is a neuropod cell that has been put in culture with a sensory neuron, and after 22 hours you can see that they have completely recapitulated the circuitry, and essentially is 2 brains in a dish. So this was the basis of a first article that we published in 2015, in which we documented that these sensory enteroendocrine cells form synapses. Since then, other laboratories have expanded upon those findings and also find that other enteroendocrine cells form synapses and form coupled to sensory neural pathways to transduce the stimuli from the lumen of the intestine or colon. We also track this connectivity, and for that we relied on monosynaptic rabies tracing. So rabies infects neurons, and what I often describe as a leap of faith experiment, we put a bolus of this virus inside of the lumen of the colon, and we're hoping to identify any cell type that will get infected. And I said that it was a leap of faith for 2 reasons. One is because we knew that the cells had some neuronal features. And 2 is that we started in the colon intentionally because it gave us, by doing an enema, it gave us a clear open access into the lumen of the colon, as opposed to having to do surgery into the mice and having the chances of side effects. And we found that not only rabies infects the cells, but when we enable the rabies to travel upstream of the circuitry, the rabies label neurons of the vagal nodose ganglia, as you can see here. So these are cells that are 1 synapse away from the lumen of the intestine. And of course, upstream of that, they end in the nucleus tractus solitarius. So this circuitry brought up 2 main points. One was that the gut, like the nose or like the tongue or like the ear or even the eye or any other epithelial surface, it has the anatomical features to be able to transduce a sense. And the second thing was that inadvertently, by using rabies, we stumbled upon a path that pathogens in the gut could use to access the central nervous system. And as you can imagine, that is of importance because several pathogens have been described to cause neurodegenerative diseases. And while they are thought to start in the periphery, it is unknown how they access the central nervous system. And of course, this circuitry offers a direct path for those pathogens. So this was the basis of another article which we published in 2018 and essentially formed the basis, the neural basis of this new sense for the gut to be able to transduce a sense to the brain. And at the same time, we also in collaboration with Professor Ivan de Araujo, his laboratory found that activation of these intestinal innervating vagal neurons was actually sufficient to induce sustained self-stimulation. And this is known as a hallmark feature of reward. So not only we eat for two reasons, one is because we need, and the other one is because it feels good. And the circuitry seems to be involved in both aspects of eating, which is one, to regulate the amount of food that we eat, but also to regulate how good it feels to eat. And just to give you a little bit of to cover a little bit of the functional experiments of how we have characterized the circuitry and what it has brought us to study the behaviors that are associated with sensory circuitry, I will touch on 3 quick experiments. So this is a vagal cough recording. And it's a rather simple technique. It's a classic technique. It uses 2 electrical wires, pretty much, that are wrapped around the cervical vagus, while a stimulus is delivered within the lumen of the intestine or the colon. So when we perfuse a stimuli in the lumen of the intestine, we get a very rapid vagal response to sugar. However, using optogenetics, in this case specifically halorhodopsin, that has been engineered in these neuropod cells, when we silence the neuropod cells, the vagus does not respond to sugars. And in fact, we have detailed the molecular basis of that, and now it is clear that the vagus actually depends on these neuropod cells to be able to distinguish not only a stimuli from sugar, but also from sweeteners. Another thing that we noticed at that time, it was that if we block the classical hormonal actions that are elicited by nutrients, we still get this very fast vagal response. So as you can see here, after 120 seconds, if we ablate we pharmacologically inhibit receptors for cholecystokinin, we abolish the response to sucrose. But the first 120 seconds remain pretty much intact. So this suggested that there has to be another mechanism that is much faster and, of course, compatible with synaptic neurotransmission to be able to transduce and also provide continuity to the sensations in the gastrointestinal tract. And through some collaborative work with some friends at Rockefeller and Harvard University, they developed a system, David Hildebrand and Professor Wei-Chun Lee at Harvard, they developed a system to be able to acquire automatically transmission electron micrographs. And this is about 2 millimeters piece of colonic tissue of a mouse that has been imaged in a tile of 35,000 images. And you will see how beautiful this is, because as you are able, as we go deeper and deeper and deeper, we can take a very close look at the cells and their organelles. And quickly here you can realize that these cells, these neuropod cells, not only they have those large dense core vesicles to secrete neuropeptides, but they also have these small clear synaptic vesicles. And indeed, in subsequent experiments, we found that those small clear synaptic vesicles they actually hold glutamate. So here is just showing the vesicular glutamate expression. And in other experiments, we have shown that these cells not only are able to package glutamate, but actually use glutamate to transduce a stimuli from sugars onto the vagus nerve. So essentially, the gut has circuitry to conduct a stimuli very rapidly to the brain, so the brain is able to adjust decisions. And what are those decisions? So this is where technology has really helped us. And to be able to answer this question, we had to rely on two different types of technologies, and we had to bring them together, as you will see. So one is optogenetics. And this is a classic video from a beautiful experiment done in 2013, I believe, by the laboratory of Professor Garrett Stubert, in which these mice are sated, but when the hypothalamic neurons that are controlling appetite are stimulated, the mouse becomes hungry, forgets that it's already sated, and starts to eat again. So it's just highlighting that optogenetics is a phenomenal technology to be able to activate specific cells within neural circuitry to be able to interrogate behaviors, right? And the second one was we needed the ability to be able to determine food choices in real time while the animal is being provided with an option of sugar versus sweetener. And for that, we relied on the PhenoMaster. And the PhenoMaster has these multiple sensors that allow to provide multiple different types of foods or other stimuli to the animal. So in this case, if we provide two types of solutions, we are able to record every half a second the responses from the mice. And then, of course, we can measure body weight and also locomotor activity. So when we give the animal the option between sugar and sweetener, the animal very rapidly, within a couple of minutes, I'll say, the animal learns which one is, the animal distinguishes. It does not learn. The animal already knows these are animals that have already been exposed to be able to stabilize the sugar preference over time. But once the animal knows, it only takes it a couple of minutes to distinguish which one is only sweetener and which one is the caloric sugar, as you can see here. But then the final point was, how do we manipulate these gut sensations while we interrogate behavior? And for this, we had to adapt gut optogenetics to the gut. Optogenetics to the gut. And one issue was that the fibers that are used in the brain, they are not flexible. They are actually rigid, as you will see here in these videos. So we had to engineer a flexible fiber that when it goes into the lumen of the intestine, it will be compatible with the delicate intestinal lumen and will not puncture the lumen of that. So when we mount and we deliver, in this way we can deliver the light to a specific region of the intestine. And the animal, as you will see here in this video, the animal is pretty happy among its little friends. So now, in this way, when we silence these neuropod cells, what is fascinating is that the animal is incapable of distinguishing the sugar from the sweetener. Essentially, revealing that these neuropod cells are to sugar in the intestine what retinal cone cells are to light wavelength or color in the eye. And furthermore, if we do inhibit pharmacologically both ionotropic and metabotropic glutamate receptors, we also see the effect, suggesting that it is the glutamatergic neurotransmission from the neuropod cells in the intestine that allows the animal to detect the sugar and then recognize the sugar from sweetener. So with that, I would like to end by saying that behind every bright brain is a wise gut. And I would like to highlight that here at Duke, we jump-started in 2014 an organization which is called now Gastronauts. And among other things, it has a podcast and also a symposium. And we have a symposium coming up next year. Look us up, thinkgastronauts.com at Nantes. And the symposium will be on May 11 to 13. And there are still a couple of spots for registration. So with that, of course, I would like to thank my team and all of you for your attention. Thank you.
Excellent. Thanks so much, Diego. And now I'd like to introduce our next presenter, Dr. Zhen Yan. Zhen, thanks so much for being with us today, and the floor is yours whenever you're ready.
I'd like to thank Liam for the introduction, and particularly I'd like to thank American Physiological Society, Inside Scientific, and the TSE for organizing and supporting this event. Today, I'd like to share with you a story about a novel voluntary weightlifting model in mice for resistant exercise. It is very well known regular exercise promotes health and prevent disease. Exercise is the best intervention to prevent, treat many diseases that we talked about today. Cardiovascular, metabolic, neurodegenerative disease, mental disease, cancer, etc. And this meta-analysis done in 2010 showing that cardiovascular fitness, which is also known as endurance exercise capacity, is the best capacity or best predictor for all cause of mortality. And fortunately, exercise is the only way to truly improve exercise capacity. So exercise itself is a predictor, and exercise training is a way to improve the predictor or the exercise capacity. The big question is that the underlying mechanism for the exercise training induced adaptation and the health benefits. We still know very little about them. According to ACSM and CDC, it is recommended that we exercise 3 or 4 times a week with a total at minimum 150 minutes of moderate to intense exercise with the endurance type of mode, but combined with 2 times per week resistance training. The reason to have combination of endurance and resistance exercise is that these 2 modes of exercise stimulate different pathways in our body, and to fully benefit from regular exercise, it is recommended to do both. And the endurance exercise, which require longer duration and stimulate the metabolic pathways, including remodeling of mitochondria, etc. On the other hand, resistant exercise promotes anabolic pathway, which will lead to hypertrophy and improved strength. To have models of 2 different modes of exercise is very, very important. One model that used to recapitulate endurance exercise is this voluntary wheel running. Naive mice run about 7 kilometers per night on the first day. If you put them into the cage, they only run during the night. As you can see from the middle top panel, that in the dark cycle, they have running activity. But on the right side of this middle top panel, you'll see that after 4 weeks of training, these mice ran significantly more. And we have 4 mice here. As a result of this 4 week of running, you can see on the right side top panel, immunofluorescence staining of plantaris muscle, which is recruited during voluntary wheel running. You see significant increase in the number of the blue fibers. These are the type 2A fibers stained with the anti-myosin heavy chain 2A. In the middle panel at the bottom, consistent with the immunofluorescence data, you'll see the significant increase in myosin heavy chain 2A protein expression with very subtle reduction of the type 2B myosin heavy chain protein. No appearance of type 1 myosin heavy chain protein. Concurrent with the increase in myosin heavy chain 2A, we observed increased PGC1 protein, a master gene for metabolic mitochondrial adaptation. This model is also known to be sufficient to induce acute response in naive animals. As shown on the right side bottom panel, you see this in response to either 3-hour running or 3H, or run for 12 hours overnight, or 3 hours post, 6, 12, 24 hours post. You can see there is an acute transient response of PGC1 mRNA and the VEGF mRNA, responsible for mitochondrial adaptation and angiogenesis. However, the model for resistant exercise is lagging behind. As you can see from this slide, I searched the PubMed and to search for a number of publications using volunteer wheel running comparing to ladder climbing, which is one of the best physiological resistance exercise model. You can see here around 2002, the volunteer wheel running model started to take off, increase exponentially, and to the level of about 450 publications per year. And the ladder climbing publication is like 1/10 of that. There are many models present for us to take advantage to study resistant exercise. In this review paper, they listed about 13 different models with a red arrow point to the models that I have personally involved. Synergistic ablation is the one remove, either cut off the tendon or remove part of the synergistic muscle to put load on the muscle, panel A and the panel B. And electrical stimulation to elicit contraction against the load in the panel E. And then we have the panel F with the bird with wind to hand weight on the wind. There's one model that is very close to the model that I'm talking about today is the panel C. Dr. Kligar published the paper in 1985 using this model to train rats to push the lever to get the food. And then the panel D showed the ladder climbing model. So these models are very, very useful for us to study resistant exercise training. However, there are limitations that prevent us from using these models to have large-scale studies. The first limitation is the invasiveness. The synergistic ablation, nerve stimulation, etc., can cause unwanted inflammatory responses in the recruited muscles. As shown in this slide, showing this synergistic ablation, I personally had that experience too, that showing massive inflammation in the peripheral region of the muscle with apoptosis. The middle panel shows the ladder climbing. We all know this is a very labor-intensive. A student or a postdoc or a technician in the lab to spend significant amount of time to train mice or rats to climb the ladder. So we cannot do large-scale study with this model, even though it's very physiological. On the right panel, it shows the difference between the normal resistant exercise in humans versus some of these frequently used model of synergistic ablation. You can see here that with 10 weeks of weightlifting, we have about 8 to 12% increase in muscle mass based on the MRI imaging analysis. At the bottom, two studies done by colleagues, Dr. Holmberg and Goodyears, showing that synergistic ablation within a short period of 2 weeks induced 50 to 100% hypertrophy. And remember that synergistic ablation causes muscle overloaded 24/7. So this amount of hypertrophy will probably never shown in humans. So pointing to the significant difference between the animal model and human adaptation. So with that in mind, we started to wonder whether we can use the food as incentive and develop the cage top to monitor and induce resistant type of exercise. I started with a shoebox and duct tape, and to put together a cage top and put the mouse inside, and the mouse will push this green color lever with the attachment to the plate, and with a magnetic sensor as shown in the white color, and a magnet on top attached to the lever. So when the mouse push this lever plate with a hole in the middle to receive the food on top, they will have to perform squat type of activity. So as shown on the right side panel, as you can see that this is the very first time we recorded their activity of pushing this lever. So with that, we were very excited about this because it shows that it's possible that to use this model, we don't have to be there to train them. So we modify the design, and finally we made a prototype and also add a component to this, which is the collar. We put this collar around the neck of the animal, which will not cause harm to their neck, and is loosely around the neck, but small enough to restrict them from climbing out of the cage through that hole, but also put the weight during the lifting on the shoulder. A key parameter for this is the height of the adjustable ramp to the food. The distance has to be less, 0.7 centimeter less than the distance from the nose to the heel of the animal. By doing that, we can calibrate the cage top and put the weight on the end of the lever and calibrate to calculate how much load put on the shoulder. With this model, we tested, and eventually we developed 3D printing for the cage top, as shown in this figure, to show that on the top we have a food container with a metal wire to prevent food from dropping, and then there's one view from the back of the cage top, and a side view showing the lever extending outside, and we can put the weight on the end of the lever. And the adjustable ramp is in the cage. So this is a video showing this mouse is pushing during the squat-like activity with the extension of the heel and to reach the food, and the weight was attached to the end of the lever through a pulley. In order to demonstrate that this is a model to recapitulate resistant exercise, we performed one single bar overnight weightlifting for these mice. Based on the recording, we observed about 420 times on the average for 9 animals, and they pushed against 150% body weight. Immediately after the overnight weightlifting activity, we harvested gastrocnemius muscle, plantaris muscle, quadriceps, TA muscle for analysis. As shown in the middle panel with RT-PCR, we saw significant increase in DSCR1, FIM14, NR4A3, but no significant increase in TWEAK, cytochrome B, mRNA. These increase in the mRNA suggest that the response to the weightlifting activity is specific to some of the genes, but not all. And then to validate that this only relevant to the recruited muscle, we measured some of the mRNA, DSCR1, FIM14 in the quadriceps, TA, heart, and liver, and compared the responsiveness. As you can see here, the increase in the quadriceps is very significant for DSCR1 and the FIM14, and no increase in TA muscle, moderate increase in the heart for the FIM14, and no increase in the liver. This data suggests that the transcriptional response only occurred in the recruited muscle, maybe a little bit increase in the heart, but not in the antagonist muscle TA for this squat-like activity. We performed RNA-seq for many genes, and we saw significant upregulation for 341 genes and the downregulation for about 150 genes, and some of them are among the genes we tested with the RT-PCR, for example, nuclear receptor 4A3 and TNF-RSF12A, which is also the name for FIM14. CFOS is another immediate early genes that you can see here. CFOS is upregulated. So with that, we decided to move on to test whether long-term weightlifting training will improve muscle function and metabolism. We developed a protocol for the training. The first 3 days, we have acclimatization. Day 1, we have this level up and the ramp up. So the mice did not have to push any weight. They did not have to lift the heel to get the food. So we call it up, up. The second day, we lowered the lever with the ramp still elevated. So we called it up, down. So this time, they had to push the lever plate, but there was no weight attached to the lever. Day 3, we lowered the lever, but also lowered the ramp. So they had to do the heel lift to get the food, but there was no additional weight on them. After those 3 days, we have them into the staging phase of the training. This will be 5 days a week, so we will allow them to do the weightlifting activity for 3 days, give them a break, and then another 2 days, another break. So during this phase, we'll start with 100% body weight load on their shoulder with the weight put on the end of the lever, and with a 20% increment every training day until they reach 240% body weight load. And then the rest of the training phase for the 8 weeks, we have 5 days a week at 240% body weight constantly. So this is the data showing that after 8 weeks of training, as you can see on the left top panel, we have the weightlifting activity. They dropped dramatically as we increase the load. They had about 200 times per night stably when we reached the level of 240% body weight load. In the middle panel on the top, we have the MRI imaging showing the increase in the plantar flexor muscles after 8 weeks of training, with about a 14% increase in cross-sectional area. We harvest the muscle, we measure different muscles, and here we show there is a significant increase in GA muscle weight, but not in TA muscle. Plantaris muscle, soleus muscle all increased in the range between 8 to 14%. At the end of the experiment, we harvest the muscle, but before we harvest muscle, we inject puromycin 30 minutes before the harvesting, and we measure the incorporation of puromycin as an indication of protein synthesis. As you can see here, there is significant increase in puromycin incorporation in the weightlifting mice compared to the sedentary control. And similarly, we saw the increase in quadriceps, plantaris muscle, moderate increase in the heart, but no increase in the liver. So these findings suggest that training promote hypertrophy along with enhanced protein synthesis. We also try to understand the signaling mechanism underlying the hypertrophy and the increased protein synthesis. We measured molecules in the mTOR pathway. As you can see here, we show a moderate increase in AKT phosphorylation with increased total AKT very moderately, and increased Raptor, S6K with the phosphor form and a total S6K. For EBP, similar result. These data suggest that there is a moderate increase in phosphorylation of AKT, slight increase in total AKT, increase in Raptor, and no increase in the phosphor S6K, but increase in the total S6K. Because of the increase in total S6K and increase for EBP1, the phosphor to the total ratio did not change. These findings suggest that there is an enhanced expression, generally speaking, there is an enhanced expression for all these proteins in the mTOR pathway. Consistent with this result, we found in the literature published by our colleague Christian in Switzerland, showing that synergistic ablation or overload model with enhanced expression of these molecules along with increased phosphorylation. So if you calculate the ratio of phosphor for EBP1, phosphor AKT, and the phosphor S6K to the total, the increase either would be moderate or no increase. So this data is very consistent with our findings with this more physiological model. We also measured contractile function using a roller system. We anesthetized the mice. We performed this plantar flexor muscle function analysis. As you can see here, the torque generated during a tetanic contraction or the relaxation speed moderately increased, but are consistently observed from different animals. But we did not see significant change in the phosphorylation curve or the fatigability protocol, as you can see here in the panel on the right on the top. Other than elevated torque generation, there is no significant shift in the pattern. We also subject the mice to treadmill running and measure the lactate level and their running distance. There's no significant increase in the exhaustive running test result with a consistent increase in lactate level in the blood. So this data suggests that this voluntary weightlifting is not inducing endurance type of adaptation. Consistent with this functional data, we measured protein expression for electron transport chain proteins and the myosin heavy chain isoform 1, 2A, and the 2B. Consistent with the functional data, we saw no significant increase of any of the mitochondrial complex proteins, nor we did see any changes in the myosin heavy chain, suggesting there is no significant increase in the slow or oxidative type of fiber. When we subject those mice to running, to weightlifting, we saw no significant drop in muscle weight, in the body weight, but there is a slight drop in the body weight in the sedentary control animal, suggesting isolation of them into a single cage caused some type of stress, but a weightlifting model seems to mitigate that. We did not see significant changes in body composition, either by measurement of fat mass, lean body mass, free water, and total water content. But we did see a moderate drop in the epididymal fat weight. We saw a very profound improvement of metabolism. As you can see here in the right panels, top panels, showing that the blood glucose after a single bolus of injection of glucose into the animal increased dramatically in the wild-type animal to a level about 300 milligram per deciliter, and followed by a clearance curve. But this increase in blood glucose is significantly reduced to a level to about 140, and with reduced level afterwards. If you calculate the area on the curve, there is a significant reduction in the area on the curve in the weight training animal. Consistent with this whole body glucose metabolism measurement, we collect the muscle before we harvest and after we harvest. In between, we inject insulin at 5 units per kilogram, and then we measure the AKT phosphorylation as a response to insulin as insulin signaling. As you can see here, at the baseline in the control animal, we did not see much of a phosphorylation of AKT, but with the weightlifting, there is a moderate increase in AKT phosphorylation. In response to insulin injection in the control animal, there is a significant increase in AKT phosphorylation, but this response of it is much more profound in the weightlifting trained mice. This data suggests that weightlifting significantly enhanced insulin signaling in skeletal muscle. This may explain the significant improvement of glucose tolerance tests. Finally, we would like to know whether this enhanced metabolism and insulin signaling is related to a process that is known to be important for clearance of the environment, improve insulin sensitivity, which is autophagy. We measured GLUT4 content. We did not see significant increase in GLUT4 content after training, suggesting the increased insulin sensitivity and glucose metabolism is not due to enhanced GLUT4. But in the same samples, we measured the P62, saw significant reduction of P62, the top band here of the two bands here, and we saw increased total LC3 with the LC31 and the LC32. But if you calculate the LC32 to LC31 ratio, there is a significant reduction. The combination of the result of P62 LC3 suggesting an enhanced autophagy flux in this weightlifting trained mice in the skeletal muscle. There was no increase in ATG7 or ATG6 protein level in skeletal muscle, suggesting that enhanced autophagy flux is regulated at the P62 LC3 level. So with that, I'd like you to have this take-home message. There is an urgent need for a model of physiological resistant exercise for large-scale studies. We developed a novel model of voluntary weightlifting cage top for mice that does not require human handling during the experiment. With this model, an acute bout of weightlifting activity elicits robust transcriptional responses only in the recruited skeletal muscles. And long-term training promotes hypertrophy, increased contraction with enhanced strength and the speed of contraction, along with enhanced expression of the mTOR pathway and the protein synthesis. Long-term weightlifting training also promotes whole-body glucose metabolism with enhanced muscle insulin sensitivity and autophagy. So we believe this model will add to the research repertoire for us to choose this model as a mode of resistant exercise. We can combine this with a volunteer wheel running to really have better understanding of endurance exercise and the resistant exercise, the underlying molecular mechanism, so we can take advantage of the information we get to develop better therapeutics. So our lab, and then I hope many more labs, will take advantage of these models to really understand the molecular mechanism of exercise-induced adaptation and their benefit in promoting health and prevent disease. And our lab has used these models to study many different disease prevention process. With that, I would like to thank our lab, particularly the person who spearheaded the study, Dr. Di Che, who is assistant professor at the Hunan University now, and a student, Becky Wilson, who is a postdoc at Duke, and Josh Drake, who is assistant professor at the Virginia Tech now, and RJ, Robert, and Mei Zhang, a technician in the lab, who all have contributed significantly to this study. And of course, I'd like to thank my founding agent, NIH, and my collaborators at UVA, and my lab members for the study. With that, I'm looking forward to hearing your questions and answering your questions. Thank you very much.
Excellent. Thanks so much, Zhen, for the fantastic presentation. So to kick things off, here's an interesting question for Diego. Does the gut have memory?
In response to stimuli, so let me break down this question in two parts. One is every cell has memory. They have to be able to remember things that they have been exposed to. And as a consequence, the gut as a whole, as a collection of cells, must be able to remember the stimuli that has been exposed to. The basis, the biology basis behind this is really unexplored. But I suspect by studying the circuitry that is connecting the gut to the brain, we are going to be able to understand a little bit more, not only about how it is that the gut remembers stimuli, but how it is that that memory of the stimuli drives previously stored behaviors that are, previously stored information that is stored in the brain to drive certain behaviors.
Yeah, great answer, Diego. Zhen, now a question for you. How reproducible are your results? It seems like quite a repetitive exercise that maybe mimics endurance. Are your results pretty consistent?
My answer to this reproducibility is that yes, our result is quite consistent. We are, almost like every day we're doing it, and we have more and more data to show that this, particularly for the glucose tolerance test and insulin sensitivity, this model is far more, profound. Even we had a direct comparison with endurance running. Weightlifting is very powerful in improving insulin sensitivity. And but I would say I still have limited experience with this, and as time goes, I think we'll get more information.
Excellent. And Zhen, just as an extension of that, have you noticed much of a difference in terms of results when you compare your original methodology to your more refined current methods?
Yeah, the shoebox data is just a proof of principle. I wanted to know whether mice will try to get there and push at all. We did not put a weight on the shoebox, and we did not measure anything other than try to see whether mice will behave that way. So there was no comparison with any results.
Excellent. Thanks, Zhen. Diego, question for you. How would you describe the interplay between the gut microbiome and neuropod cells?
That is a very interesting question, and it's the subject of active exploration. In fact, these cells do have receptors that allow them to recognize not only microbial metabolites, but microbial patterns. How it is that they are able to discern these stimuli to guide behavior still remains unknown, but you can imagine that they provide a direct conduit to be able to, for the animal to be able to recognize the presence or absence of certain bacterial ligands, to be able to adjust its behavior, not only locally at the organ level, like increased gut motility, but also at the whole organism level by increasing or decreasing appetite or desires for specific nutrients.
Excellent. Great answer. And I think in the interest of time, we'll make this next question the last one. Zhen, it's an interesting question for you. Do mice eat less if they have to exercise to feed, or do they actually eat more because they need to use up some energy exerting themselves to get at the food? And also, do the mice gain weight over the training period?
That's an excellent question. We we did pay attention to that. We also were asked by the reviewers when we try to publish the paper. The answer is that they ate almost the same amount as the control animals. And if we place the food and the device is in the right position, and we had no difference in them. If you notice that in one of the figures, we show that when we put mice in the individual cages, the control mice start to lose a little bit of body weight. But in the weight trained mice, when we put into the cage, somehow because they have this weightlifting activity, they gain a little bit of body weight. So I would say there's no profound difference between the control and the weightlifting mice if you put them on the normal chow diet. There is no significant difference in the amount of food they ate during the night.
Excellent, great answer. So thanks so much, Zhen and Diego, for all the fantastic insights today, both in your presentations as well as the Q&A session.
Well, thank you, and thank you, audience.
And in closing, thank you again for taking part in this Inside Scientific webinar produced in partnership with the American Physiological Society, and we look forward to having you with us again soon. Have a wonderful day, everyone.